Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT JRF Entrance Exam question paper conducted in the year 2023. If you are new to my channel, please do like, share and subscribe. Then next one. Which of the following disease is characterized by predominant motor dysfunction that results from the progressive degeneration of the nigrostriatal dopamine pathway? So it is Parkinson's disease. Answer is option 1. Then which of the following is true about pairwise alignment of protein sequences? Local pairwise alignment can be performed using Needleman-Wunsch algorithm. It is not Needleman, it is Smith-Waterman algorithm. Local pairwise alignment can be performed using Smith-Waterman algorithm. Answer is option 2. Local pairwise alignment may be performed using Smith-Waterman algorithms. Then next one. Given below are two statements. Statement 1. In a neuron, standard resting state is minus 70 MV. Treatment with the chemical X induced intracellular potential minus 50 MV, while when treated with another chemical Y, it showed minus 90 MV. Given such condition, we can say X induced depolarization while Y induced hyperpolarization. Statement 2. In a neuron at standard resting state of minus 70 MV, treatment with the chemical X induced intracellular potential minus 50 MV, while when treated with another chemical Y, it showed minus 90 MV. Given such a condition, we can say Y induced depolarization while X induced hyperpolarization. Okay. Now what is the first statement? In neuron at standard resting potential minus 70 MV, treatment with X induced intracellular potential of minus 50 MV. So X is causing the resting membrane potential to become less negative, that is from minus 70 MV to minus 50 MV. So it is depolarization. So as per statement 1, we can say X induced depolarization. As per statement 2, it is hyperpolarization. So statement 2 is not correct while statement 1 is correct. And Y showed minus 90 MV. So it is causing the resting membrane potential to become more negative. That is from minus 70 MV to minus 90 MV. So it is hyperpolarization. So the answer here is statement 1 is correct, but statement 2 is incorrect. Answer is option 3. Then next one. Which of the following statements about Foster resonance energy transfer FRET is incorrect? The energy of the excited molecule donor passes directly to a nearby molecule, the acceptor. That is correct. The energy transfer is possible only when the donor and acceptor are close to each other within 50 angstrom. That is correct. The efficiency of FRET is inversely proportional to the sixth power of the distance between donor and acceptor. That is also correct. The efficiency of FRET is directly proportional to the sixth power of the, it is not directly proportional, it is inversely proportional. So they have asked here which is the incorrect statement. So answer is option 4. Then which of the following is produced commercially by mammalian cell culture? So it is tissue plasminogen activator. Answer is option 2. Then next one. Which gene is generally inserted in an adenoviral vector to treat cancer by suicide gene therapy? So suicide gene therapy, it is based on the you deliver a cytotoxic protein, a gene encoding a cytotoxic protein into tumor cells. So this cytotoxic protein, it will be producing cytotoxic proteins which will cause the death of the tumor cell. That is suicide gene therapy. Now, which gene is generally inserted in adenoviral vector to treat cancer by suicide gene therapy, they have asked. So it is HSV-TK. HSV-TK is herpes simplex virus thymidine kinase gene. Now what does this gene do? It converts the antiviral drug, that is ganciclovir, to a monophosphorylated molecule. This monophosphorylated molecule, it is then metabolized to a toxic triphosphate by certain cellular kinases. So it will result in the production of toxic compounds, thereby inhibiting DNA synthesis and producing death of the tumor cells. Next question. Which of the following will generally not occur if the function of myosin 1 and myosin 2 is blocked? So cell crawling will not occur. Answer is option 2. Then next one. Recombinant interferon gamma is commercially produced by cell culture of. So it is produced by peripheral lymphocyte cells. Answer is option 3. A duplex DNA sequence that read identically on both the strands is known as a palindrome. Next one. Hemolytic disease of the newborn develops when maternal IgG antibodies specific for fetal blood group antigen cross the plasma and destroy fetal red blood cell. The disease is called erythroblastosis fetalis. Answer is option 1. Then next one. Which one of the following is the drug of choice for sexually transmitted disease caused by chlamydia trachomatis? So it is azithromycin. Answer is option 2. Next question. Match list 1 with list 2. Ajmalcin, Rauwolfia serpentina, A3. Anethol, Ilium verum, B1. Codeine, Papaver somniferum, C2. Vincristine catranthus roseus. So A3, B1, C2, D4. Answer is option 2. Next one. A co-integrate vector is used to transfer cloned genes into which one of the following? So co-integrate vector, it is used in plant cell genetic engineering. So answer is option 2. Next one. Human genome contains approximately 25,000 genes, but the number of proteins in human cell is estimated to be approximately 250,000. Which of the following can explain this difference? So human gene, it contains only 25,000 genes, but it codes for 250,000 proteins. Now why is it so? So it is because of alternative splicing and post-translational modification. Answer is option 4. Then next one. Podophyllitoxin. Match the list 1 and list 2. List 1 is phenolic compounds and list 2 is the plant source. So podophyllitoxin is mayapple. Curcumin is turmeric rhizome. Methyl leguanol is basil and anethol is star anise. So the answer here is A1, B3, C4 and D2. Answer is option 3. The binding site on an antibody is known as. So epitope is present on the antigen, paratope is on the antibody. So they have asked for binding site on an antibody. It is called a paratope. Answer is option 1. Blossom scoring matrix is used for. So it is used for alignment of protein sequences only. Answer is option 3. Coming to the next question. Chemically, what is milk of magnesia? Milk of magnesia is magnesium hydroxide. Then next one. Match the type of chromatographic technique with the protein property. Dye affinity, structure and hydrophobicity, chromatofocusing, charge and isoelectric point, covalent thiol groups, gel filtration, molecular size. So the answer here is A3, B1, C4, D2. Answer is option 1. Dash may survive without the need for immunosuppression. Corneal grafts, that is for your eyes. So answer is option 2. Then next one. An alternate form of starch biosynthesis machinery in cereal endosperm involved. So it involves cytosolic ADP glucose pyrophosphorylase. Answer is option 2. Then next one. Genetically male sterile plants can be developed by overexpressing the Barnase gene. Answer is option 3. Then next one. Mammalian cell cultures are difficult to scale up in conventional bioreactors for the production of therapeutic proteins primarily because high shear sensitivity. Answer is option 2. Match the animal cell lines with their commercial application. List 2. Baby hamster kidney cells, foot and mouth vaccine, myeloma cell lines, monoclonal antibodies, simian kidney epithelial cells, polio vaccine, porcine kidney cells, urokinase. So the answer here is A4, B1, C3, D2. Answer is option 1. Then what contributes most towards the hydrophobic effect when a protein is folding at room temperature? Entropy of the water molecules. Answer is option 1. Then next one. The expression of which of the following types of cyclins in plant is modulated by growth factors such as cytokinins, auxin, brasinosteroids, sucrose and gibberellins? So it is D type. Answer is option. Which statistical test is used to calculate false discovery rate? Benjamini-Hochberg. Answer is option 1. Then next one. Which of the following enzymes add an extra adenine at the 3' end of the DNA strand? It is Taq DNA polymerase. It is used in PCR. After PCR, the 3' end of the DNA will have an extra adenine, and this property of this particular Taq DNA polymerase is used for TA cloning vectors. So the answer is option 2. Then next one. Which of these animals have recurrence of estrus events during their breeding seasons? It is squirrels. Answer is option 4. Next one. Myasthenia gravis is primarily caused due to defects in the cholinergic receptor system. Answer is option 3. Then match list 1 with list 2. List 1 marine alga, list 2 type of alga. Ficus serratus, brown algae, Gellidium, red algae, Chlorella vulgaris, green algae, Cyanophyceae, blue-green algae. So A1, B2, C3, D4. Answer is option 2. Then which one of the following terms is used to describe the protection of the unimmunized individual based on immunization of a sufficient number of other members of the population? It is herd immunity. We have seen this in case of corona infection. Then next one. 2 protein sequences can be aligned with the help of a blossom matrix. The blossom matrix were developed by aligning sequences of distantly related sequences, not closely related sequences but distantly related sequences. So answer is option 2. Then next one. Given below are 2 statements. One is labeled as assertion A and another is labeled as reason R. Assertion A. Threading is a de novo protein structure prediction method suitable for frequency with very low similarity. Sequence with very low similarities. That is correct. The threading method is more efficient than homology modeling. That is not correct because homology modeling is more effective when a close homologous template is available, while protein threading can provide insights when no close homologous templates are found. So A is correct but R is not correct. So the answer here is option 3. Next one. An integral plasma membrane protein is glycosylated. Scientists can trace the journey of this protein from the time of its initial translation to its translocation to the plasma membrane in its mature form. Which of the following changes in its molecular weight can be expected to take place during its maturation? So first there will be an initial decrease followed by an increase, because initially it will not be glycosylated but later it will be glycosylated, so the molecular weight is going to increase. So first it will decrease, then it will increase. Then next one. Which of the following United Nations Sustainable Development Goals is related to clean water and sanitation for all? SGD 6, SDG 6. Answer is option 3. Which of the following combination of cytokines represent only isoprenoid cytokines? Cis-cysteine and dihydrocysteine. Answer is option 3. Then next one. Which of the following fragments regions of an antibody molecule will you use to identify the presence of a specific epitope in a biological sample? FV, FC, CH, DL. Now this is your structure of the immunoglobulin. This is your Fab region. VL, this one. This is your Fab region. This is your FC region. And this is your FV region. Variable region of the light chain, variable region of the heavy chain. They have asked which region of an antibody molecule will you use to identify the presence of a specific epitope. Epitope is present in the antigen. So the epitope will bind to the paratope of the antibody. So you have to see for the FV region. Answer is option 1. Because FV region is having the paratope, which will bind to the epitope of the antigen. Then match list 1 with list 2. A lipid anchor for proteins attached with the membrane. GPI. 3 is the answer. Only A4 is given in 3. Then ribonucleoprotein complex to bind to the signal peptide. SRP. Signal recognition particle found in, I think, in endoplasmic reticulum. Then formation of correct disulfide bonds in proteins. PDI. Chaperone in the endoplasmic reticulum lumen. BIP. So the answer here is A4, B1, C2, B3, D3. Answer is option 3. Conversion of organic matter to biogas proceeds in 4 stages. Hydrolysis, acetogenesis and methanogenesis. Which one of the following microorganisms play key role in the methanogenesis conversion stage? That is archaea. Archaea is having the methanogens. So the answer here is option 3. Then next one. Given below are 2 statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. A shuttle vector can be used in 2 different host organisms. That is correct. A shuttle vector contains only one origin of replication. No, a shuttle vector has 2 origin of replication which help the plasmid to replicate in different host organisms. So R is not correct. So the answer here is A is true but R is false. Answer is option 3. Next one. Given below are 2 statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. Termination of DNA synthesis occurs upon incorporation of a nucleotide lacking hydroxyl group on the 3' carbon of the pentose sugar into the growing DNA chain. Okay, that is correct. The hydroxyl group on the 3' carbon of the pentose sugar is involved in the phosphodiester bond formation. That is also correct, and that is the reason for this particular assertion. So both A and R are true, and R is the correct explanation of A. Answer is option 1. Among the following, the feature that does not contribute to host tropism in bacterial pathogenesis is. So host tropism is that certain pathogens, they have specificity for a particular host or host tissues. That is called as host tropism. So which does not contribute to host tropism is body temperature of the host. Answer is option 3. Which stage of mitosis is blocked by colchicine? It is metaphase. Answer is option 4. Then match list 1 with list 2. S phase of cell division. DNA of parent cell is replicated. G2 phase. New proteins are synthesized and the cell approximately doubles in size. M phase. 2 daughter cells are produced and G0 is the quiescent phase. So A2, B3, C4 and D1. That is option 4. This was a very easy match the following. If you know the various cell division cycle stages, then it is easier for you to answer this one. So that's it. Thank you for watching my video. If you are new to my channel, please do like, share and subscribe. I have uploaded other previous year question paper with the solutions and explanation for the solution I have answered with the previous year question papers. Some from which year I don't know. I have uploaded a few years question papers of DBT. As well as CSIR also I have uploaded. You can go through it. Thank you for watching my channel.